fly through them. And in case you have questions, let's wait, until after the presentation, so we won't go over time. So my name is Nayereh Ghoreishi. I am the DTC manager. I'm representing the developmental therapeutics core today. That's a core that has been established since 2012. We are part of the Chemistry of Life Processes Institute. So we are located in Evanston. So we've been around for a long, long time. The core provides fee-for-service preclinical and translational support to all NU investigators, also external investigators. And we serve both Evanston and Chicago campuses. So as I said, the mission statement of the core is to rapidly and efficiently advance your novel therapeutics from basic research to the clinic by providing preclinical and translational support and expertise. So DTC doesn't have any instruments. We are not an instrument-based. How we charge you for services is like for our expertise, in animal models or in vitro models. So no equipment, and I don't have any, fancy movies, demonstrating what we do in that sense. This is the org chart for DTC. I, along with Venon, we report to Dr. Shannon Lee. And I have Elizabeth Dempsey and Grace Ma, who are both research associate, and they report to me. We are hiring a new full-time employee that's been posted since February with specific expertise to run PDX models. So if you guys are interested, or if you know anybody who's interested, please have them come and talk to me. And then Ivana Stepien has been in the course since 2017. So she's, older than I am at the core. I joined the core in 2020. So I just celebrated my 3rd year anniversary. And Ivana is a core specialist, and she basically, does a lot of hands-on tasks at the core. So I'm here to talk about drug development, mostly for you guys. As you know, drug development is a very extensive, very costly process. It takes average of 10 years for a compound to find its way into the market, and the cost is in the billions. So the average cost, I put about 2.6 billion. So what DTC comes into play is mostly in the preclinical area for you guys. So that's really earliest stage after your basic, research discovery part. That's where you come to my core for specific studies. What happened? Okay, all right. So there are numerous cores through Northwestern that could help you with these various stages of your drug development. Where we come into play again is later on, maybe in the screening and hits to lead. If you want to look at, you want to do in vivo experiments and you want to look at mechanism of action, efficacy, or later, if you want to optimize your lead, you want to evaluate it in terms of toxicology, pharmacokinetics, again, efficacy. So the experiments that we do is basically IND enabling you. So in order for you to push your drug into the clinic, you have to have a package that you submit to FDA, and you have to have specific a characterization of your compound. Have you done this assay, that assay, evaluated pharmacokinetics, toxicology, show me efficacy, show me dose response, time courses. So we help you put that package together before you apply for IND. So as I said, one of the expertise that we provide is pharmacokinetics and biodistribution, okay? Another one is preliminary tox studies. So FDA requires long-term tox studies, eventually in dogs and in rats. But what we do is like the basic, tox studies for you in rodents and for shorter duration. We also provide cancer research biology. And if you have a specific model, you want to bring it in, my core has the expertise to do that. We also have ICOG protocols, and we could do immunization and antibody production, both in mice and in rabbits, and help you with your animal protocol write-up. So DTC already has 9 blank animal protocols. In order for you to perform any animal studies, you need to have, because animal work is regulated under ACUC, the Animal Care and Use Committee. So you have to have an animal protocol or an amendment to an existing active animal protocol. So we help you with your animal protocol, and then we perform the studies from A to Z, starting with the animal procurement and then ordering supplies and running the experiments, etc. Other expertise that my core has is surgical expertise. For surgeries, you have to have aseptic techniques, you have to have isoflurane machine, you have to have like the know-how if you're doing injectable anesthesia. So we are experts in performing a lot of surgical procedures. We also do necropsy. If you're interested in collecting organs for various reasons, we help you. If you need to perfuse the organs, we have the capability to do that. If you're evaluating your bloods, in your study, let's say for blood, let's say for toxicity, you want to evaluate the blood chemistry or for blood count, we have the capability to do that. We also have a stereotach device, so we could do not only drug administration, but also if you're interested in glioma, for example, we could do tumor inoculation both in rats and mice. Other supports are drug administration. We do various routes, the routine routes, PO, IV, IP, subcutaneous. We've done cancer experiments that they wanted to do the drug intratumorally. We've done that. Or let's say you have a compound that, it's very short-lived and you want to do as a mini pump. Or if, for example, let's say for your cancer biology, if you do a compound that, let's say, has a short half-life, right? So you dose animal in the morning and, by 10 hours, the compound is cleared into the system while the tumor cells are growing. So it's ideal if you are, you have a compound to do azet mini pumps. We have azet mini pumps that you could implant subcutaneously or in the peritoneal cavity, and it could go for 4 weeks, maximum 4 weeks. So your drug is in the system at specific rate, at constant rate, for a long period of time. So that's something to consider. We also help you with the study design. It's really important to think twice, measure twice, and cut once, right? Animal experiments are very expensive. It's not only the cost of animals, and the process is very lengthy. It takes a long time for animal amendments to go through. As I said, they're regulated. So not only they're time consuming, but they're very expensive. Cost of the animals, husbandry, shipping, ordering them, procurement charges. So it's really good to think about your experiment very thoroughly and come up with a design that you get the maximum endpoint or data from those experiments, right? And then we also, once we run an experiment, we collect data, we give you interim data. If you want to, one of the things that's nice working with the core, for example, compared to, let's say, going to a CRO, we're local. Our mission is to help a new investigator move forward with their drugs and research. So let's say in the interim, you decided, okay, this dose is too high and it's showing adverse events. And if you want to modify it, the core is not like a CRO. The CRO says, no, you've done that. We're not changing it. This is what your contract is. But the nice thing about, coming to the core is that we work with you guys. We want you to succeed. So you could come to us and modify your experiments, right? We work with you. So, our mission is to help you guys move forward with your therapeutics. And also we help with manuscript and grant writing, So I'm going to talk about pharmacokinetics studies. Again, you have a compound, you want to profile that compound in terms of absorption, distribution, metabolism, and excretion, right? So it's really important. And the rodent PK, there are formulas that you could translate that into human PK. So one of the first phases of clinical trial is phase 1. You have healthy volunteers and you want to dose the compound and look at adverse events. So by running a rodent PK, you could translate into the patients, right? So you dose a compound, you take blood at various time points, and you profile that. How is your compound getting into the system? What is the Cmax? How long the compound stays in your system? What is the T half-life? And then if you're interested, let's see, you want to see if your compound is reaching the site of action. You're evaluating, for example, glioma, right? So you need the compound to get into the brain. We could collect brain and you could analyze, the brain sample to see if your compound is in the brain or not. Or, you want to evaluate how is your compound getting excreted and, leaving the system. Is it through kidney? Is it through liver? Is it through lungs? So we could collect those tissues and, analyze for the compound. So it's really important to have a PK experiment. Also, as I said, for dose and dose frequency, the T-half will tell you if your compound is clearing, quickly, or we've had compounds that stayed in the system for over a week, right? So you don't need to dose, the compound daily or twice a day or whatever. You could just dose the compound once a day. So it's very informative to have that kind of data before you embark into more expensive efficacy experiments. So 10% of the total cost of the preclinical study is compared to 90% of for clinical development or development, only 10% of it is for preclinical. So it's not as much as the rest of drug development compound. But it's interesting that 80% of the compound that go in the preclinical testing, they fail. And most of them fail because there's a poor pharmacokinetic effects of the compound. And about 30% of them fail because there are lack of efficacy. 11% of them fail because of toxicology effects. So it's important, again, I want to drill that into you that the PK is really important. It'll give you the half-life of the compound. It'll give you the Cmax. It tells you whether your compound metabolizes and whether it metabolizes into active or inactive compound. Is it the active metabolized that is, inducing the effect, or is it the whole compound? So it gives you ideas about that. And again, whether your compound crosses blood-brain barrier, not only whether, that's like a site of action, for glioma and other like neuropathic pain or other, you do want your drug to get into the brain, but getting your compound for other therapeutics into the brain might mean that there would be CNS, toxicity. So it's always a good piece of data to have. So things to consider in relate to PK study. The first thing is your compound. What is the dose that you want to test in the PK? If you don't know that, it would be nice to just maybe do a dose response, do a couple of doses and see, whether you see the compound in the system or not. How many doses do you want to do? Would it be one dose or multiple doses to make sure that the drug gets into the system? And the route is really important. Are you going to have oral, administration, IP administration, sub-Q administration, IV? Also, the solubility of your compound. If you're doing an IV injection in mice, you better make sure that your compound is soluble, right? Otherwise, it won't go through, it clog the needle. So the compound is like doing a null study. The compound won't get into the system. And even though, let's say IV or IP, routes might not be translational, But, you're doing a dirty PK to profile your compound. Eventually, once you have profiled your compound and, you realize that you get a good PK profile, you get a good efficacy, then you could modify the SAR to change that so it's more translational into the clinic, right? And then the vehicle. Most compounds go in 100% DMSO, but in terms of the iCook, you can't have those 100% DMSO pushed into the animals. The allowable concentration is 10%. So vehicle is also very important, and we help you formulate your compound. We have a list of what compounds, what solvents, what concentration is allowable, and help you formulate it so you have a soluble compound for PK. Also stability. If your compound goes in the solution, but by the time you take it to vivarium and it crashes out, it's not a good thing. Also in terms of solubility, for oral administration, if your compound is like in a very fine suspensions, you could get away with it, but not with the IV or IP route. So that's something to bear in mind. And then in terms of the animal, you want to figure out what species you want to use, what strain, you want to use rats or mice, what kind of strain inbred mice or outbred mice, what sex, what age, and for number again. So for number, if your route of administration is orally, you could get away with 3. We've had screening compounds for, a project, and there has been hundreds of compounds, and we just do 3 compounds orally and you get the PK profile. But if you want to do IV, I suggest to do larger N. It's much more tedious and harder to do, mouse tail vein IV injection. So I recommend to do 4 or 5, just in case, the needle moves and the compound doesn't get in the system or the needle comes out of the vein. So it's better to cover your base and, do the experiment right. And again, how many doses you need to do before your PK. Do you want to do a PK in naive mice or diseased mice? We've done PK analysis, further down in line, they wanted to evaluate, like for tumor-bearing mice, does the compound gets into the tumor? So you might want to do tumor-bearing mice, for your PK. But most of the time, if you're early on, in the early stages, we recommend just doing a naive PK. But pick a strain that correspond to the background strain of your efficacy model, right? And then the volume of the blood is really important. And that depends on the method of analysis, right? Are you going to do a ELISA assay? For ELISA assay, you don't need much serum. You need, maybe 25 microliter. But if you're sending your blood to IMSERC for mass spec, they require at least 500 microliters. So again, those things you have to consider, what do you want to do? And also, I suggest if you run a PK, make sure you collect tissue. As I said, animal experiments are very expensive, so it's better to collect the tissue, even if you don't have the funds right now to analyze it, freeze it for later analysis. And if you move forward and get more funds, you could always go back and analyze those tissue. So there are 2 different models that we do at DTC for PK. One is serial bleeding and one is terminal bleeding. And there's advantages and disadvantages to both models, right? For serial bleeding, you are using the same mice, you're dosing them with a compound, and you're bleeding them via tail vein at different time points, right? So you are using less animals and you're using less compound. But the disadvantage is because you're doing serial bleeding of the same mice, you're limited with the volume of the blood that you could draw. Again, that's something that there are maximum blood drawn allowable by the ACUC. So, we can't, take loads and loads of blood. You have to be mindful and have that approved, right? And then if your compound is IV injected, you do need, and you're taking blood from your tail vein, you do need a good tail, right? We've had compounds that had been run in the PK and they were, hemolytic compound. And that sort of shuts down the tailwind. So we can't do serial bleeding with those kind of compounds. So these are things that, you need to consider. And also, if you're evaluating your, biodistribution, because we're keeping these mice alive for a long period of time, we are sacrificing them only at the end. So you could have like organ biodistribution at the end time point, right? For terminal bleeding, however, you get different cohorts, 3 mice per different time points. So you order a whole bunch more animal. And so you could do tissue biodistribution at every time point because we are stacking those mice, at 1 hour, 2 hour, 4 hours. So we have like 3 mice per time point. And therefore, because we're euthanizing the mice, and we're bleeding via cardiac puncture, you get a whole lot more blood, right? But the disadvantage is that you're looking at the metabolism in different cohorts of animal over time point. But most of the time that doesn't matter because you're using very homogeneous strains. And, if your technique is good, they correlate. And of course, because you're sacrificing different mice at different time points, you're using more animals, and therefore you have to have more compounds. So these are the considerations that you have to bear in mind running a PK study. This is an example that we did in collaboration with Dr. Kalapurakal here at NU and Pfizer. So PK study, not only is translational based on the PK profile that you could figure out what doses, clinicians need for patient, but you could also back translate it, right? So these, we had, 3 different CDK4/6 inhibitors that are already in the market: ribociclib, palbociclib, and abemaciclib. So they were already in the clinic, and Pfizer wanted to know what is, the PK in mice. Interestingly, that wasn't done. So we dosed the mice for 4 days, and then we collected blood, and we sent the blood samples to Pfizer. And these are, they correlated the human versus mouse PK in these graphs, and, send that to us. So that was a nice study that we did. So this one is a PK that we did with Dr. Gianneschi. He has, they evaluated paclitaxel. They're using human serum albumin to deliver paclitaxel. So they had this VTX, which is 5 complexes of ODDA with paclitaxel compared to Abraxane or paclitaxel in Cremophore. And they did that in healthy mice versus tumor mice. So, and they evaluated the PK profile at different time, doses. And then they also looked at the biodistribution in the blood, in the tumor and versus liver. So those are the right graphs. So you could do fancy, analysis as well in the PK. So now I'm going to move on into toxicity and safety assessment. That's one of the services that we provide. So Paracelsus, the father of modern toxicity, said that all things are poisonous, only the dose makes it non-poisonous. So all chemicals, synthetic or natural, they have capacity to be toxic. And I put that table in, for example, aspirin. Aspirin has a therapeutic dose and has a toxic dose. So it could be with any drugs. You have a therapeutic window and you have a toxic, dose. So the dose is the key concept in toxicology, right? As I said, we provide preliminary tox studies for you, both acute and subchronic, to evaluate the maximum tolerated dose and also evaluate the adverse events, after the animals are being dosed with your compound. So the consideration again, strain, there is a strain variability, there is gender variability, age variability, and whether it's naive or diseased animals, you get a different tox profile, right? And also bear in mind that, when we do acute MTD for that one, for example, you dose a mouse with a set of concentration, let's say 100 mg per kg, and you watch them for an hour. And if they don't show any adverse event, you escalate the dose to 200. And then you're not seeing any adverse event, you escalate it to 300. And then you titrate back, at 300, you see some, adverse event, you titrate it back. So that's how and these are different cohorts of mice that we use, right? So we could go up to 6 doses. Usually that's how many doses you can test until we figure out your maximum tolerated dose. But okay, so maximum tolerated dose is an acute dose. It's dosed only once. But you have your compound and you want to do daily administration because you're doing a disease model. You want to dose daily until like the tumor is regressed or doesn't get enlarged. So that MTD, if you're dosing in a disease, paradigm, might become LD, might become lethal because of the poor metabolism or maybe because of accumulation. So these are the things that you have to consider when you are using your MTD and you want to translate it into your efficacy. Maybe you should cut that down and maybe make it, or before you test it in your efficacy, maybe do a chronic dosing. Dose it for 7 days, dose it for 14 days, collect the organ and evaluate it microscopically and see what you see, So this is an example of subchronic tox study that we do. Again, we dose them for 7 days, and then we observe the animals, twice a day, and we look at the weight. Body weight is really interesting. A lot of times, the toxicity, is shown in the loss of body weight. But at the end, we also collect, all the organs and the blood, and we could send it to, usually send it to MHPL, for histology. And then we have a preclinical pathologist that actually he works for Charles River, but he does pro bono work for us, and he evaluates those slides, the H&E slides from different organs, and he scores them, and we usually have a meeting with you and him, and he could tell you about the abnormalities that he's seen. We also have capabilities to collect urine and stool if you're interested in like how the compound is metabolized. We could also do that for you. The behavior adverse events that we're looking at, it could be, are your mouse aware? Are they alert? Are they passive? How is their mood? Are they grooming? If usually, rodents, when they vocalize, that's a sign of pain, right? Are they reactive? When you touch them, how are they showing reactivity? We also look at autonomic responses, respiration, heart rates, writing, CNS responses. You could look at their gait, do they have the more motor coordinations? How is their muscle tones? You could do a grip test, and see if, they have a good posture. Or, if they're convulsing, twitching, that means that you have CNS, toxicity. So now we get to cancer research biology. And again, as I said, we have the protocols, we do animal procurement, we inoculate the mice, and then we could do their drug administration and monitor body weight and tumor growth through this study for you. DTC has about over 100 cell lines, immortalized cell lines. We could not only expand them for you, but we also sell those cell lines if you want. We make sure they are viable, and we could sell them cheaper than if you buy them from ATTC. We also perform proliferation and apoptosis assay. In cancer research biology, the first thing is for you to do is the cytotoxicity. First show me in the cancer cell lines, maybe NCI, 60 panel, or the cancer cell line of your interest, if your compound, shows cytotoxicity, and what is the IC50 for that, right? And then we could do efficacy model, for you guys. We do have some syngeneic mouse models, and, that are, the cell lines are murine derived. Unfortunately, that's very limited, but we do have those models. We have performed them. We do xenograft whether it's heterotopic or orthotopic. We've worked with humanized mice from Charles River and Jackson Labs. We've done studies in metastatic, and we also use IBIS imaging. We use CAMIS IBIS imaging. So if you're, let's say, cell line or your compound is, tagged with bioluminescence, we could also do those studies, imaging studies for you. So here, I'm not going to go through it. There are cell lines and organoids. There are advantages and disadvantages of, both things. Cell lines are cheap. It's easy. You could do them, but they're very homogeneous. They're only, bidimensional, and they don't represent all the, complexities of like a cancer or tumor, right? But with the organoids, they are 3D, they are more translational, they're heterogeneous. You could use them for high throughput, but the drawback is that they don't have the inter-organ communication, they don't have the immune component, and they're not, not all tumors, you can't grow organoids from all tumors, right? And here, again, I talked about the cancer models that we have. The considerations that you have to have in selecting your mouse model is, first of all, figure out what are you targeting? What is your therapeutic aim, right? Are you going after immunotherapy? Then you have to choose the proper model, right? What is your tumor property? Are you going after solid tumor or blood-derived tumor, right? Then the study paradigm. What's your growth, tumor growth rate? What is the treatment duration? And whether those models are available, how long it takes to make those models, and what is the cost? If you have a limited fund, you have to choose your model very wisely. We suggest at DTC to definitely run a pilot study before you do your efficacy experiment. And the reason is for you to find out your tumor growth rate and your therapeutic windows. If you have a very aggressive cell line and you're doing 6 million cell inoculation, and before you know it, your tumor has popped to 200, millimeter cube, your efficacy window is very short. So you want to maybe titer it down, run a pilot and titer the cell number down. So the tumors will grow slower, but that would give you enough time for your compound to have that therapeutic window, right? So these are the things that you have to consider when you want to choose a model. And then, eventually, let's say you have a compound, it shows efficacy in a cancer model. What about if like the cancer cells are dormant? Maybe after you stop, dosing, you want to keep those animals and monitor them. Does the cancer come back, right? So dormancies, metastasis, or, if your compound, let's say it's not as efficacious, but what if you combine it with adjuvant therapy, radiation, dissection, or other, would your compound have added benefits? So these are things that you could think about later on. And, all the vendors, Charles River, Taconic, Jackson Lab, they have extensive tables about, what kind of animal model, there are, what are their costs, what you should use, so I'm not going to go through this. There's nude mice, skid mice, NSG mice, NRG mice. And, depending on, again, your therapeutics, are you going after lymphocyte myeloma, or leukemia, you might want to choose a, different kind of model. Or do you want to just establish, PDX? You have, working with an oncologist and you have primary tumor and you want to establish that, as a PDX, and you choose an NSG. But if you're, just want to see, if your compound is effective, you might just be able to, and you're going after solid tumors, you might just be able to get away with nude mice, right, or skid mice, instead of, getting more expensive NSG mice. So it all depends on your therapeutics, what you're after, what kind of cancer you're going after, and then what kind of mice you should order. And here I have listed the price for the, some of the mice. And as you can see, that the prices go up as you get fancier. If, you're lacking T cells, B cells, and natural killer cells, the price of NSG is much higher. If you're interested in humanized mice, which is, a great model, and it's more translational to study, for example, blood-derived cancers, because you're studying immune system, and it's good to have the human immune system in a mouse. So those models become much more expensive. If you're looking at the PBMC model, it's the cost is 480. We've done a model actually for Dr. Josh Leonard, and they use CD34 mice, female mice. And the cost was 1,000, and we did an N of 10, and that was like $10,000 just for the cost of mice, right? So depends on where you are and what you want to do. Choose your model wisely, because animals are expensive. Here I'm showing you again, we have a lot of cell lines. That's an image of, adherent cell and suspension cell. We could validate them, expand them, sell them to you, or run, in vitro assays for you guys. This was some viability tests that we did, proliferation assays. The first two graphs are done for Dr. Green, and he tested two different compounds, DCA and CA in mouse and human hepatocytes, and compare them. The right one was a protease inhibitor, RMD-102, that we did for Dr. Shimomura for RMD Science Incorporation, and his aim was myeloma. So we tested the compound in two different cell lines, OPM2 and K562, and as you can see in the table, the IC50 was lower for K562. So that's, the model that they aimed after. Now we're getting into the in vivo expertise, and this was an orthopedic patient-derived model that we did. These were kidney cancer cells that we implanted orthotopically in the kidney capsule right from the patient. And then once it grew, we expanded it in the second generation mice. This work was done with collaboration of CAMI. We have nice MRI images, and the circle is the cancer growing on the kidney. This other example was done with Dr. Abdul Kadir. So for his MYC inhibitor, and I believe they used SCID mice. We did the tumor inoculation. They did, tumor volume for different doses. We also did just a naive CD1 mice. They wanted to look at the toxicology. So we looked at the clinical sign, and as you can see, we tested various doses, 50 mg, 200, 220, 240. We didn't see any tox or adverse events, in the general appearance, conditioning, behavior, or posture, or respiration, or skin and hair color. But once we got to a higher dose, 250 mg, we saw adverse events, right? And then from the same mice, they collected organs, and they evaluated with the histology. So spleen, lung, liver, kidney, heart, and brain were evaluated. Okay, this was the orthotopic and subcutaneous cancer that model that was done for Dr. Shilatifar and Panos, and I believe they used NSG mice, and this was a proof of concept model that they did. So evaluating USP7 role in the Notch pathway, and they used short hair pin RNA, and knocked down the compound, or they had an inhibitor, and then we did IBIS and evaluated on day 7, day 21. We generated the survival graph, tumor volume, etc. This one also was a follow-up on the same pathway done by Dr. Foltz's lab. They used JORCAT and COT-LL, and the short hair RNA to evaluate that. They knocked down the gene, and they evaluated, the tumor volume and survival after doing that. These are all published. This work was done with Dr. Mandilo. He used human shock factor 1 and 2, knocked down version of the mice, and again evaluated it in prostate cancer, I believe. And they looked at tumor volume growth. They also harvested the tumors, and they did RNA-seq, in collaboration with the core here. And they also looked at the tumor histology, H&E, and also with the, looking at the expression of HSF1 and F2. This work, the lower part of it, I showed you before with Dr. Gianeschi. This was done in nude mice, and again was with VTX. And so we had also, you could see the subcutaneous tumors, and those pictures are showing day 1 versus day 21, and they're showing the tumor in the vehicle group, at day 1 and versus day 21. This work was done with Dr. Huang and with her meta-arrestin compound, and that was in a PDX and NSG mice. And again, this was, I believe, organoid driven, and we generated, we performed the experiment, we generated the tumor volume, the tumor weight, and the body weight for Dr. Huang. So, okay, how do we measure tumor volume? So usually, if it's a subcutaneous, right, we use a caliper. It's been, a way of measuring tumor volumes. You plug it in the formula, you get the tumor volume. But, this formula assumes that your tumor is ellipsoid, right? Because you don't measure the height. And the drawback is a lot of times you have a necrotic tumor, or your tumor is deep down, into the tissue. So the tumor volume might not be as accurate, right? And also there's a lot of bias. We usually allocate the same tech for the same study to measure tumor volume, because there is, the investigator variability. If I go measure tumor volume, depending on how I restrain the mice and how tight I put the calipers, around the tumor, I might get a different value than if my technician, who has done it for a lot longer, gets those tumor values. So but having said that, if she measured it in the morning versus in the afternoon, her values, even though not significantly, might be different. So there's both inter and inter, investigator differences. But having said that, this is a way that a lot of pharmaceutical companies have used it for decades to, get to tumor volume. They publish, they push the drugs into the market. However, things have changed. The technology has advanced, and now we have automated tumor volume analyzers. So I'm proud to say that we, through the equipment grant from Office for Research, we were awarded funds to purchase Biopticon, which is a automated tumor imager and tumor manager. It's a beautiful, it's got a beautiful software. It accurately measures the tumor volume very quickly, improves p-value. It's fast. You could operate it with hand or hand-free. There's no sedation, is needed. It's real-time data analysis, and the tumor is regardless of the tumor shape. If your tumor, let's say, is, it's got multiple lobes or is necrotic, it just gets rid of all those, factors. So we're very happy with that. We just started getting trained, and we are using it actually this week on a new experiment that we are doing with Tempest. And the program, you could do 50 groups, you could do 100 animals per group, and 20 drugs, in one study. So it's really a great equipment. We're very happy to have it. And again, the software produces graphs. You could monitor, like, for example, on the right side, it's like, I believe animal number 88 is highlighted, and you could see the tumor progression with time. So it's really, a great software. It's a great, piece of equipment. And also you could generate a template, and then, once you start scanning, the data accumulates, and then it does also statistical analysis. For example, if one of your tumors is very small, or the body weight is very small from day one, before you allocate or divide your group, randomize your group, it says, this is an outlier, you shouldn't keep it, in your study. So it's a very comprehensive and a great piece of equipment. So surgical expertise, as I said, we have a stereotactic device. We could do orthotopic, inoculation for you. If you want to do as a mini pump, we have capability of that. Or if your cancer, a mechanism of action is bioangiogenesis, we have a rabbit ocular model. We could do that for you as well. This is the DTC workflow. You come to me with your study request, and, we do a few back and forth, because the more I know about your study scope, what kind of, cell line, is it organoid, is it cell line? How many cell lines have you done your pilot? Do you know your tumor growth factor? So we go a few back and forth, and we generate a study plan. And then I calculate a cost estimation, and you say, oh, no, my grant is just for, let's say 10,000, and I can't afford, doing this fancy experiment. And we could cut it down, but cut it down in a way that still your money is not wasted and you're getting to your endpoint, right? Once, we agree on the cost estimation, we submit a ICOG addendum, and that's where the bottle neck is. So it takes about 6 weeks for an amendment to get approved, because you submit it to ICOG, they send it to multiple reviewers, and the reviewers come with questions, clarify this, why are you doing this, why are you doing that? So we go back and forth, a couple of times with the reviewers, but once it gets approved, we could order the animals for you, expand the cell lines, purchase consumable, etc. We generate the data, and then we work also, let's say you're interested in imaging, you want to do PET scan, with CAMI, we coordinate, for MRI and, all the imaging with other cores. If you're doing a PK, we coordinate with MCER. If you are interested in metabolomics, for example, we could cut the tumors and send it to metabolomics core, RNA-seq core. So, we work with them, we work with external CROs. So we do from A to Z for you. The last few slides that I have is just like listing what kind of, tissue cell lines we have and what they are. I could send you the link. We are in process of updating our website. It is on the website, but we have, multiple cell lines. If you are interested in the PDXs, I have a list that Dr. Renan Kang has generated. I could tell you, unfortunately, with the PDX, it's very time consuming to establish those PDXs, and there are not that many available, but we do have some. Here are the list of cell lines from pharynx, prostate, skin. If you're interested in breast cancer, metastatic breast cancer, if you're interested in myeloma, leukemia, lymphocyte, ovary, etc. This list is a murine drive, non-human cell lines. Again, a lot of, cancer, I say drugs, it's good to have a syngeneic model, right? Because with the syngeneic model, you have an intact immune system. And I was working at Abbott, and like the Abbott PARP inhibitor that came into the market, I was doing the glioblastoma for that project, which was, we were using syngeneic model. So it's a good thing to have, but again, it depends on your therapeutic. If you want to do PDX and it's a human, grafted into the mouse, you do want to have immunodeficient mice. So, but we do have some murine drive cell lines as well. So with that, I'm closing my talk. Sorry, I talked too much, but if you have any questions, sure. Thank you.